To RTP 180. So for those who are here for the first time, thank you for playing along with that. We are coming up on almost going into like the 4th year doing this. And I've done every single one of them. And I say this, thank you. Wow, yes. So either I don't have a life or I really enjoy doing it, and I do enjoy doing it, so I have good news and bad news for you. Good news is I'm going to do this again next year. Bad news is I'm doing it again next year. So first timers, hang in there with us. We like to have a little bit of fun. We're going to have a great time this evening. So welcome. Thanks for coming, and let's kick this thing off. Make sure this is on. There it is. So for all of you who are here for the first time, my name is Will, and I have just a small little marketing company here in Raleigh in the Research Triangle Park. Ooh, we've got fancy transitions. Love it. You are in the RTP Frontier building. So if you are not familiar with this building, first time in it, this whole first level is kind of an open co-working area during the day. So if you need a place to plop down, have a meeting, get some work done away from the other co-workers, a great spot that is free all day long to come in, plop down. These chairs are gone. There's desks and tables and a lot of cool people that are working out of here. So please feel free to drop in and say hello and work out of here. All right, tonight, we have 3 presenters this evening, a little shy of what we normally do. So they will get 5 minutes, but we're gonna give them a little leeway there. I lied to you in the back there, I'm sorry. I am gonna ding the triangle just so you know that that is the 5-minute mark, so that I'm over here doing this to get your attention. I will ding the triangle at 5 minutes. Feel free to continue, just let that be a warning that you've got another minute or 2. Don't go another 10. We've had somebody do that. We've had somebody go 15 minutes before. Don't break that record this evening. Then we will open it up for 2 questions, and they will answer those questions. And then because we only have 3 this evening, we will then bring all 3 of them up at the end in case you have a question and it did not get answered. They will come up and we will do a 7 to 10 minute little panel at the very end. What'd I tell you? See, you have to do some magic to make it there up. All right. We'd love for you to be social, but do it online. This room is very hot room, so no floor, no ceiling. If you are chatting off to the side or to your friend, just text them or tweet them. You can tweet us at the RTP. You can tweet using hashtag RTP180. There it goes. And of course, please leave the place as you found it this evening. RTP is very gracious to let us use and give us the space to do this. So please just make sure you clean up after yourselves before leaving. And I kind of already mentioned this, everyone can hear you, so just make sure that you are not talking during the presentations. They've poured their lives into their research, and we would love for them to have a nice, quiet, and attentive audience this evening. And I think, yes, that is all the rules and regulations. Who's pumped? Who's ready? All right, okay, lukewarm. I get it. All right, let Anna do what? You need a joke?
Yeah.
Nothing like putting me on the spot there. You know, I researched drug development corny jokes, and there really wasn't much that came up. Surprising, I know. I'll think of one. We'll get through the first. I don't want them to have to follow the joke, you know? Too good. All right, I'm gonna let Anna continue to get this rolling, make sure we're good. I think we should be good. All set. All right, first up. Alex Dusek is the head of commercial strategy at the Durham-based biotech Argos Therapeutics, charged with leading the launch strategy for their late-stage investigational product AGS-003 that has the potential to be the world's first truly individualized precision immunotherapy for cancer. Throughout his career, he has cultivated a passion for the potential of personalized medicine to transform how we treat life-threatening diseases. Ladies and gentlemen, please help me welcome Alex to the stage.
Thank you. Well, thank you. It's my pleasure to be here on behalf of Argos Therapeutics, a Durham-based biotech company. And as Will mentioned, we're developing the first, hopefully, launching the first precision immunotherapy for oncology, for cancer. So I want to start out tonight with this slide, and the theme of this talk is we're all different, and everybody's different. In medicine, however, I don't think we've made enough progress in treating everybody differently. There we go. This is a quote from a local professor, Dr. Ralph Snyderman from Duke. He actually just joined the Argos board. And the basic premise here is one size doesn't fit all. And so there's a lot of technology and a lot of data that has been emerging in the ability to amass big data and also genetic sequencing to really think about personalized medicine as a wave of the future. The White House has a huge initiative on precision medicine, and that's their term for it, and we're going to be using that term tonight, precision medicine. So what is imprecise about current medicine? Well, for those of you who aren't in drug development, this is sort of how it works. On the left side is a trial. So you have a placebo or an active drug, right? And then you test it on patients, and you sort of bucket people into response or non-response, positive outcomes or negative outcomes. And if, on average, your treatment does better than placebo, then you can get the drug approved. But that's on average, right? So you can see the gray patients there. They're more in the positive outcome. The white patients there, a little bit more in the negative outcome. But it's not completely homogeneous. There's some people in gray who have negative outcomes and some people in white who have positive. So then you kind of amplify that potential error, and when the drug's launched and FDA approved, prescribers prescribe it, and 75% of the patients don't respond. Okay? And so what precision medicine is trying to ask the question, why? Why do those patients respond and why do those patients not respond? So, as Will mentioned, one of the interests I've had, this is a paper that I co-authored last year with some folks from Duke on biomarkers in a different therapeutic area, but biomarkers. What's a biomarker? So it's plasma or tissue. It's something from your body that goes further to identify what's unique about your disease. So, for instance, you can have kidney cancer, but this person's kidney cancer is very different from that person's kidney cancer. And that's the premise here is what type of biomarkers exist such that you can identify what's unique about that disease? That's one step. The next step is who's going to interpret that? So you have to have an interpretation, and you say, well, that's different. Let's say it's a mutation. You do a genetic test. What's the norm? What's the database that establishes, is it different? Is it relevant? Is it a driver mutation or is it a passenger mutation? That interpretation is key. The second issue is, even if you do have a biomarker that can phenotype you into, let's say here, just for the sake of argument, yellow, green, and purple, your unique disease, the only thing that matters is whether that biomarker can lead to a precise treatment. And we've made some advances in that. Here you see a genetic test for the patient with a mutation that's yellow, and they have a targeted treatment that matches that biomarker. But we're very far away from having biomarkers for all of these treatments. And matched treatments and matched biomarkers is a huge obstacle that still exists. How many people have heard about immuno-oncology? Yeah. That's basically having the patient, your body fight your cancer. So people have cancer every day. but the immune system fights it off. And what immuno-oncology is harnessing the body's own natural immune system to fight off that disease. It's very different than traditional medicine like chemotherapy or radiation therapy, where it's sort of imprecise, or even drug-targeted therapy, which uses pills, but it's kind of indiscriminate. You have collateral damage. The immuno-oncology field is taking the immune system that exists currently and revving it up, harnessing your own power to fight your own disease. Problems with immune therapy is if it's blanketed and it's not precise, you will have off-target damage or collateral damage. And so some of these therapies, recently, there are a lot of publications out of challenges with toxicities. And I think the third speaker tonight, we're going to talk a little bit more about that. So at Argos, we say, all right, well, if every single tumor is different, every patient's different, their disease is different, what is the key to tumor specificity? There's things called neoantigens. So these are things that are unique about that patient's tumor. And that's because all mutations are random, right? So everybody's going to have a different tumor evolving at a different mutational rate with different mutational patterns. And so we said, how can we harness neoantigens? That was well timed. This is what we do at Argos. So we basically take all the tenets of precision medicine and immuno-oncology and really go after precise immunotherapy. What do we mean by that? Well, neoimmunity without autoimmunity is generating a novel immune response, something that is new and unique to that patient's tumor without the collateral damage of it turning on normal tissue, right? So a very specific response. The two components of our technology are the dendritic cell. The dendritic cell is like the mastermind. It goes around the body and it looks for foreign antigens, whether it be a virus or a bacterium or a cancer cell. And it finds it and then it presents it to the immune system. It processes it. These antigens go to the immune system and says, go look for that. That is non-self. That's a foreign invader. The other part of our technology is mRNA or messenger RNA. It's like the thing that transmits the unique fingerprint of that tumor. So what we do is we try to combine those two things. So how do we do it? We take cells from the patients. This is called autologous cell therapy. But unlike any other company to date, we actually take the individual tumor from the patient, and we take the RNA from that tumor, and we load the dendritic cell, and we supercharge that dendritic cell, and then we give it back to the patient. And it goes back to the patient to restore their immune function and to instruct and arm their immune system to fight that specific tumor. So, sort of in sum here, the challenge here is how do you do that? How do you do it one patient, one tumor, going back to that individual patient? Think about how drugs are delivered right now. You go to a pharmacy and there's a common pill bottle that gets your name slapped on the bottle, and that's your medicine. But it's really the same medicine that other people are getting. It's kind of like saying, I don't know if you guys saw this ad campaign from Coke. It's a good campaign, but just because your name is on the bottle doesn't mean what's in it is any different, right? So what we at Argos do every day, we touch the patients. We recognize that there's a patient's cells behind that's got to go back only to that one patient who provided that material. I don't know if you can see it here, you probably can't, but it says subject ID number. And what we want to do at Argos is turn that to the patients behind it, right? We know every patient is different. We want to have Harry's precision immunotherapy, Ann's, Tom's. It's going back to that single patient. If we're successful, we're going to unleash a firestorm on that patient's cancer. Thank you.
All right. 2 questions. Anybody got a question? You have a question.
Thank you for the presentation. So
Sure.
my question is for your own prediction for the future. I wonder about your idea when the gene therapy and immunotherapy will be 100% efficiency for the next future. What do you think about the timeline? Is it going to happen in 10 years or 50 years?
It's a really good question. I was thinking about that on the way over. I think that we are making an advance here, right? We are going after something that's truly individualized, unique to that one patient. But I'm also not claiming that 100% response, right? So how do you get from something like I was showing you of 25% responders to closer to 100%? I think that's going to be really tough because there's as much as we can phenotype and genetic test patients and the technology with big data, I think that getting to 100%, there's always going to be idiopathic issues with patients that we just can't figure out. So I guess we could probably get closer to 90%, maybe in another decade, but that's probably my prediction of the time frame.
All right, one more. I'm back here. Yep, you sir. I see you on the left. Yep, creepy, huh? I know.
What are the possibilities of an autoimmune response here?
Yeah, so there are the papers that I mentioned, the New England Journal paper, the SITC paper, and a couple other reports of late with a couple of the therapies called CAR-T cell therapies. They're amazing therapies because in many patients they're a cure, at least as far as we've monitored the patients. But in others, there've been some deaths because they haven't been able to turn off the immune system. So they rev it up and then it goes too far. It's called cytokine storm syndrome. With our therapy, that's one of the hallmarks of our therapy is because it's completely autologous, meaning it's from the patient. And what we put in the dendritic cell that is actually from the patient is the tumor itself. We believe that because both the tumor source is autologous and the cell source is autologous, it's going to be minimal toxicity burden because you're not, let's say, putting a single antigen that's shared with other patients in it that's sort of indiscriminately going after all the tissue. Hopefully, if the theory is correct, and all of our phase 2 data has shown it so far as a low toxicity burden, is we're only going to be targeting the specific tissue to which that patient's dendritic cells have been armed. Thank you.
So much, Alex. All right, since the front row asked for a joke, I was able to come up with one. It's not about drug development, but it is about snowmen. Close, right? Where do snowmen keep their money? Snowbanks. Dr. Doris, you gotta follow that. I'm sorry, I really am. As Vice President of RTI's Center of Global Health, Dr. Rouse has extensive experience in the formation of public-private consortia and bringing together diverse disciplines to address global health needs. She leads collaborative drug development programs for tuberculosis and antimicrobial resistant bacteria. Dr. Doris, please come up to the stage.
So that was a very funny joke, but I've got another one. This is the season everybody's spending more time in shopping centers than you normally do. But you know, if you've seen one shopping center, you've seen them all. Thank you, it's great to be here. What a fun occasion. This is wonderful. All right.
Oh, you flip it 180. See what I did there? 180.
Good one. I'll throw another pun at you. All right, so I'm going to talk tonight about tuberculosis drug development as a case study of how a public-private partnership can be successful. And my colleague in the audience, Tony Hickey, is also working in TB drug development, so you can talk with him over the beer afterwards as well. So normally when I give my partnership talk, I show this slide first. By way of warning, but you're getting the condensed version, so this shouldn't be a problem unless you've had more than one beer tonight. So. So why public-private partnerships? What are they and why do we need them? Well, they're collaborations between industry and academia, governments and nonprofits to develop new medical products. And we need them because industry is reducing their investment, especially in early stage R&D for certain therapeutic areas. And a very good example of that is antibiotics. There are a number of companies that have gotten out of the antibiotic development area. But there are also synergies that are realized from the expertise and the resources, the facilities across the different sectors that might be involved in industry or universities and government. And there's the potential for data sharing across different clinical trials as well. So there is a need, and you'll see in the case of tuberculosis that there's a serious need for a partnership. These are some examples of public-private partnerships. There are hundreds, but I won't put you to sleep by going through all of those. But these are some examples addressing different therapeutic areas. So we'll start in on TB, and you'll understand what that is and how a partnership's been helpful. Tuberculosis is coming up. I'm sorry. Okay. It is really the oldest of human diseases that we know of. It's often called the white plague, consumption in a number of old novels, or the graveyard cough. Evidence of TB is found in human remains as long as 5,000 years ago, 5,000 BC. It's also in the writings of Homer, Aristotle, early Chinese writings. It was a leading cause of death in the mid-17th century in London. And a number of luminaries that have contributed to our society and our culture died of TB, including my favorite North Carolina author, Thomas Wolfe. So a number of you are probably out there saying, well, that's great, Doris, but that's a history lesson. We're here to learn about drug development. That's old, that's history. We don't have TB anymore. That's what I hear from a lot of people. Unfortunately, that's not the case. It is currently a serious global epidemic. 1/3 of the world is infected with tuberculosis. It's the leading cause of death from infectious disease. It's surpassed HIV now. 2 billion people in the world carry TB, and someone's infected every second. And 1.5 to 1.8 million people die every year of tuberculosis. There's no continent untouched. Treatment is 4 drugs over a 6 to 9 month period for drug-sensitive TB. But the last new drug that was developed for treatment of tuberculosis is incorporated in a regimen 50 years ago. So if that doesn't get your attention, let's move on to extremely drug-resistant TB. This is a TB that is resistant to all of those 4 standard drugs that I mentioned. So there is no real cure. People try a lot of different things. It's been confirmed in more than 100 countries. We treat people for 2 years. They have to be isolated because TB is caused by Mycobacterium tuberculosis, an infectious disease, and it's spread by coughing or sneezing, so it's very easily spread. So you have to isolate these patients because they have a very serious form of it. And after 2 years of treatment, only 16% cure rate. So XDR is emerging as an extremely deadly and costly global health threat. So you can see that it's an ideal candidate for collaboration among the different sectors on a global basis to try to find a better cure for TB. Certainly one more recent one, 50 years. And for that reason, in 2000, the Global Alliance for TB Drug Development is a nonprofit, public-private partnership formed in 2000. It involved governments, industry, donor entities, and they came together to form an entity that would address these 3 areas of their mission, to develop new and better TB drugs, obviously. But they went beyond that. And they also said, well, we can develop the best drug in the world, but it can't be accessed because it's too expensive. Or people don't incorporate it into their country policy, it won't have the impact that you would hope that it would. So they also are including ensuring affordability, access, and adoption as one of their objectives. And the third objective is not just to develop their own, but to coordinate and catalyze TB drug development by others around the world. TB Alliance participants, a number of them are shown here. The major contributors have been Bill and Melinda Gates, have been wonderful in supporting it. but also the National Institutes of Health, Rockefeller Foundation, and others. I'm from RTI International, and we were very fortunate to be part of the formation of the TB Alliance in 2000 and its ongoing operation in drug development. All right. So early on, we looked for compounds that could be incorporated into the TB Alliance for development. And we found a library of compounds with a small biotech in Seattle called Pathogenesis. And we found this particular promising compound is one of theirs, and the TB Alliance licensed it in order to develop it. And when that licensing took place, a San Francisco columnist had this to say about it. This deal tells me that biotech leaders, international health officials, and philanthropic groups are trying to create mechanisms to address needs that would otherwise fall between the cracks. I'm keeping my fingers crossed for both the TB drug and the larger experiment in nonprofit drug development. This was in the early 2000s. I want this guy to cross his fingers for any endeavor I undertake because I'm happy to say that I'm keeping you in suspense, aren't I? Come on, you can do it. All right. This drug that was licensed in the early 2000s is now in phase 3, the final phase of clinical trials before being approved by regulatory agencies for marketing. And it has demonstrated exciting potential as a safe and effective drug treatment for both drug-sensitive and drug-resistant TB. And I'll explain what the drug-resistant TB is. So this was taken forward by that public-private partnership, and it is in phase 3 and looks to be very successful. So drug-sensitive, drug-resistant TB. I mentioned it a little bit earlier when we talked about what the scope of the problem is. It is a disease in which it kills most anyone who comes in contact with it. It's a 2-year therapy. I think I mentioned earlier it's a 16% success rate. Even after 2 years of therapy, injections once a day for a month, for 6 months, pills for up to 24 months, that's 17,000 pills. And IV infusions twice a day. Toxic, expensive, very difficult to give in low-income countries. And so to address this, the TB Alliance, in collaboration with others, have initiated this Nyx Tv Trial. It's a clinical trial testing regimen with PA824, the compound we just discussed, bedaquiline and linezolid. So it's showing promise that rather than this very complex, toxic, expensive 2-year therapy, it's very promising for a 6 to 9 month therapy with only 3 to 4 pills taken every day. No injections, no infusions. So if a collaboration, a global collaboration and partnership can do this, it's very exciting. And I have a story to tell you about this trial, and I heard this story when I went to a meeting in Cape Town not too long ago, and a woman who was directing this Nyx clinical trial told the story about a young woman who had been in this 2-year therapy for 10 months, had been isolated in the hospital from her family. She turned 21 during that 10-month period, and nothing was working, and they knew that they had to do something different. And this therapy, the PA824 bedaquiline linezolid therapy, they said we have to start her on that. It's experimental, but there's no choice. So they did, and about 3 weeks later, she was still in the hospital. A worker went by her room and heard very loud sobbing coming out of that room. And they thought, well, she's having a psychic break. Maybe it's an effect of the drug. So they sent in a mental health professional to help her through it. As it turns out, she was sobbing because she felt so much better. She knew she was going to get well and she could get out to see her family. The week before that conference at Cape Town, she went home. So with successes like that, I am encouraged that in the not too distant future, we're going to hear a chorus of people around the world singing. I got to keep you singing this song. Ding dong, TB is dead. Thank you.
All right. Questions? Good time for 2.
I
all right, anybody else? I want to make sure that maybe we ask it during the panel when they come back up. Just want to make sure everybody has a chance to get involved.
Hi, thank you for a great presentation. The drug that you're working on has a nitro group, which often can be associated with idiosyncratic reactions in people, but that is often very hard to detect in people, except after a lot of people have been tested over a long period of time. Have you addressed that?
Yes, we've done, obviously, in order to get through, it's gone through the whole FDA trial approach, and as a nitroimidazole, we did look at that issue closely, and it's turned out to be safe as well as effective. So. But I'd be glad to talk with you more about any additional studies you've seen.
All right, we got one more back here.
So what are the major factors that contribute to the success of this public-private collaboration that otherwise would not have been done?
That's a great question. And we just actually finished a study for HHS to look at a number of partnerships. But the number of success factors include engaging people from all those different sectors, whether it's governments, not just US, but you saw there was Irish and DFID, and Australians and others were involved in this. Governments, user groups, patient advocacy groups, been very much involved, industry, and the not-for-profit. The not-for-profit that was established. So involving all those groups and giving all of them a voice was very important to that. And honestly, it would not have been successful, it would not have been possible were it not for those philanthropic groups like the Bill and Melinda Gates Foundation, the Rockefeller Foundation, and others. So we had to engage them and convince them that it was going to be done well. And one of the other very important things was this issue of access. The countries are really looking at for that, to make sure that if we come up with the best solution in the world, that people can actually have access to it. So we involved the ministries of health as well to find out exactly how this should be designed in order to increase this access. There are a number of other factors, setting up the right form of governance so that everyone has a say in what direction you take, setting up a strong scientific advisory panel, etc. So we've learned a lot over the years of these public-private partnerships. Thank you.
All right. I'll take the clicker back. Thank you. I feel like we would hang out. I love that joke in the beginning. We would be friends. I know we would. Best friends. Okay. She's laughing at that. That's not the joke I was going to tell. This is going to be the joke. This one's a stretch, so I know you all are smart. I know you can figure this out. All right? What do you get when you cross a Christmas tree and an iPad? Yeah? Nobody? A pineapple. She's got her head. That was pretty good. Come on. That was all right. All right. I'll quit. I do want to make mention that if you can't ever make it, we do live stream this. I don't really ever bring that up, but we live stream it every evening that we do it. And next year, we're going to beef up the live stream even more with a high definition camera and all that fun stuff. So if you cannot make it, you can watch online during that evening every month. So just wanted to mention that. All right, last but certainly not least, Dr. Alexander Tropsha, PhD, is K.H. Lee Distinguished Professor and Associate Dean of Pharmacoinformatics and Data Science at the UNC Eshelman School of Pharmacy. His research interests are in the areas of computer-assisted drug design, computational toxicology, cheminformatics, and structural bioinformatics. You really tried hard to mess me up in that bio, didn't you? I think I got pretty close. All right, ladies and gentlemen, please bring up Dr. Alexander.
Thank you for saying all those words. I was challenged by Will's opening remarks. I searched for drug development jokes. And I got a question from Google, and the question was, do you mean drug development jobs? So I don't know if it's a joke to find a job these days, but we certainly cannot rely on Google to entertain ourselves. So what I was asked to do is to talk about something as fun and as intoxicating as chemical toxicity, more specifically chemical toxicity prediction, more specifically our recent work where we have challenged some of the long-standing approaches to toxicity prediction known as structural alerts, which is this word that I will ask you to pronounce slowly later. And to put sort of everybody on the same platform, let me tell you what's meant by this and make everybody a chemist instantly. I'm sure most people have dreamt about it since you graduated from the high school. So what's meant by this is something that is very simple and very desirable to have a simple tool to predict if a chemical is toxic or not. And that is to look at a bunch of chemicals that show toxicity in some tests or assays and find out that they all share, or most of them share, a particular rather simple looking chemical group, and then based on this analysis make a statement that all chemicals that have this group are toxic, such as, for instance, nitro-containing chemicals. And with that, well, the rest is a simple story. You publish, you get the grant, you become famous, get more grants, and then entertain yourself, publish software, sell it to FDA, and become really rich and really famous. And so that's a standard story as far as how things like this are developed, and it's easy to use, and it's something that we all want, simple tools to triage compounds that are unlikely to see the day of life because they will eventually be shown to be toxic. And the company, pharma company, chemical company profits will go down. So that's what it has been. Now let's look at this molecule. And some of you may recognize its structure and some may not. But it's the best-selling drugs of all time. And so it must be a good molecule. But then we also know about the history of structural alerts, and there is a lot of software that allows us to ask if such a group, or if a toxic group, or a toxicity or structural alert is present in any given molecule. And so you just ask a question with a lot of software that's written and available, if there is anything wrong with this molecule. And there is a reference if somebody wants to execute this yourself, and you find that it has idiosyncratic toxicity, and it has non-genotoxic carcinogenicity, and it has skin sensitization potential, and it could have genotoxic carcinogenicity, etc. So it has multiple functional groups that would make this molecule totally impossible to be pursued at the very early stages of drug discovery. And that certainly creates a question as to what is possibly wrong and what could possibly go wrong, and how come one of the most selling molecules that is still on the market could make it there, and why things are as bad as they appear to be. And then you look for what the developers of software to identify such alerts are thinking about when they run into multiple cases like this. And you find a code that I think is probably one of the best I've seen, and the developers are saying the name of the software that was called skin sensitization alerts is changed to skin sensitization reactivity domain to reflect the fact that alerts provide grouping interactivity mode of action and do not predict skin sensitization potential. Now, what is something that is touted as predictive methodology, predictive software, but now is not used to predict what it's supposed to predict? So you search for answers, and I'm a chemist, but sometimes chemists need to go to quantum physicists for answers. But before, you search for a pictogram that reflects this sort of attitude, and that's the best I could find. This is how I interpret the previous statement. So going to physicists, Werner Heisenberg once said there is a fundamental error in separating the parts from the whole. The mistake of atomizing what should not be atomized. Unity and complementarity constitute reality. And that basically to say that sometimes the whole is more than the sum of its parts. And sometimes you cannot divide and conquer. Sometimes you conquer when you do not divide. And the examples coming from the world of careful analysis of chemicals, and here is the nitro group. The nitro group that's supposed to be toxic all the time. And the reality is that if you analyze, so I'm glad you asked this question, because I have an answer. The reality is that whether or not nitro group causes toxicity, or mutagenicity in this case, depends on what it's bound to. So these examples, all of them, have this nitro group, but these molecules also contain an aromatic entity, and there is a mechanism explaining that toxicity, whereas when it's bound to an alkyl group, it does not cause toxicity. And that essentially says that what matters is the whole molecule, not any individual part. So is there a methodology that actually could look at the entire molecule? And that's a long-standing methodology. Thank you for the reminder. That's an alarm. That is called quantitative structure-activity relationship modeling, that looks at the molecule as a whole, calculates something known as chemical descriptors, and then builds models that can be compared to, what else do we do? Is that's a dance of some sort. Maybe someone could push the button. Yeah. And what it does, it's a black box. That's the negative. But it does the same thing as the allures. That is that it predicts whether or not, but it does predict. It's not shy of saying, I predict whether a molecule is toxic or non-toxic as a whole without looking at individual composing allures. And so the next question, of course, is if you have alternative approaches to making predictions. One is based on something simple and transparent. And second, that's based on black box, which irritates every regulator and every biologist in the world. Which one would make more accurate predictions? So this is the experimental data about skin sensitization, and this is a comparison between allures-based system and whole molecule-based system. And of course, I chose the best example I could, but generally speaking, in multiple comparisons, this works about 50% of the time in a binary prediction experiment, and this works in about 70 to 75% of the time. And that's the result of multiple comparisons, for the reason that the whole is not the same as the part. And so then the question is, what can we do now, and what's the final outcome of this type of analysis? And the outcome is rather simple. Can we use structural errors? Yes, we can. Can we rely on them? No. Can they work? Sometimes they can. Can we know if they work or not? Yes. If on top of creating a hypothesis based on the presence of certain structural alerts, we also use a holistic model that will evaluate whether or not this alert has a value quantitatively. And so we've come up then with a consolidated strategy that integrates the old rules and the old type of development model that, for the sake of being a black box, was not popular in toxicity prediction, and demonstrated that that gives us collectively combination of alerts plus alerts we could derive from these black box models gives us a much higher prediction accuracy in a number of toxicity cases. So that's pretty much what we've done recently, and I'll probably spare you of reading. I assume most people, I'm a foreigner, if I could read English, you should too. So thank you very much.
All right, 2 questions, and then we will bring them all up for a few minutes for a few follow-up questions. Everybody got, I mean, you all understand everything we just said? Nobody has to follow up? No? You, sir? All right.
Since nobody else volunteered. The main problem with my experience with QSAR is the quality of the data that goes into the structural alerts. How do you ensure that you're getting good quality data? Because I know there are a number of these startup QSAR companies that hire biology students to review the literature, and that doesn't do very well.
Well, I shall say that this is the problem of not the QSR, it's a problem of everything. Every area of science, the data quality and data reproducibility is a problem. And for the reason, that's the reason why models often don't work. A good example is the outcome of recent elections, where all models, for the most part, failed, right? And there were statistical models that failed. So this is an issue, and we have a series of papers with a general title, Trust but Verify, where we go into protocols of how data should be curated and how the trustworthy data should be used to boost the quality of models.
One more, then I can bring all of them up.
So it looks like for a given structural alert, it's a risk factor, and there's like a certain degree as to, well, how risky is it really in this particular case? And can you put some sort of quantitation to that, so like when it crosses a certain threshold, maybe you should go through a certain standard type of test to mitigate that risk factor to say, okay, well, maybe it's not so bad or it really is bad.
So I think what we should do is to combine the science presented by two speakers before me and say that we need to develop personalized toxicity alerts and run clinical trials on them. And so it's essentially, there is a risk associated with every alert, and there is a risk associated with every chemical, and the risk is, when we come to humans, the risk is personalized, right? When we cancel drugs, it's because not everybody died, but a few patients. So that's what we need to figure out. I mean, I could talk longer about it. Yes, we need to mitigate risk.
All right, so we'll bring the other two up. Just come on up. I will grab the other two chairs so you don't have to worry about bringing chairs up here. At least you're comfortable for the next few minutes. We'll grab this one. Thank you. All right, so Ash, if you'll just do like a 10-minute timer on the phone, and we'll see where that gets us. Free for all. Ask anybody anything. He stole my question, so, all right.
Thank you. Hi, this is a question actually for everybody. Given the impact on the stock markets around the world or in the US with both presidential candidates and President-elect Trump, just curious your thoughts about what the drug industry needs to be doing to kind of support pricing and continue to be able to invest development in great drugs in the future, and what can we be doing as an industry?
That's a really, really highly charged question. I think it's very hard to predict. I think that the traditional argument is a sort of more capitalistic notion leads to greater innovation. By the same token, I think there are lots of examples of more socialized medicine success. And so I think that it's really hard to predict what Trump is going to do and what this administration will lead to. It's very, as far as all of our prediction algorithms go, I think that's the most uncertain. So that's probably not a very useful answer, but that's the best I can come up with.
It's supposed to be a fun evening, so I think the answer is everybody ages and many people develop dementia. So just invest into something that leaders of this country are likely to experience. That's safe.
I would just make the comment that drug prices are largely what they are because of the cost of drug development. It is quite a cost to take something all the way through to approval. Speaks to the need for resources to come from multiple sectors so we can drive down that cost and the risk to industry, and then perhaps the eventual price on the market would be less because they didn't have to invest as much.
All right, hold on, I'll come up so you can get a microphone.
As long as you're holding the microphone. This question is, given this public-private partnership and all the players in it, who holds the rights to the drug that you're developing? Because that will affect the cost and the distribution.
Great question. In this case, And in most of these partnerships, the TB Alliance, the nonprofit partnership, has licensed the patent from the company that developed, in that case Pathogens, and that's true for the other drugs they're developing. But what they did in that negotiation, and this was quite innovative, they said, we will pay you royalties on sales for this drug in wealthy nations. We will not pay you royalties on low-income countries because we have to meet that second mission of theirs, affordable drugs. And it was quite a tug of war to come to, between attorneys, thankfully, not myself, to come to the conclusion, but they finally agreed to it. And I think that was quite groundbreaking, and other partnerships have picked up that approach as well, in order to keep the price low in those countries that couldn't afford it otherwise.
You, sir? I believe I saw your hand.
So, yeah, Doris, if you'll keep holding onto that microphone. So, first, congratulations on apparently a tremendous success. It's quite heartening to see. But as I know, these bacteria are really pretty clever and they're very persistent. So what's your plan when resistance develops to this drug? Do you have in-class and out-of-class programs ongoing so that when the witch, rises from the dead and becomes an even worse zombie, that you're prepared?
Yeah, that's true. I think we were so fortunate to go almost 50 years without any serious drug resistant to the, and that's why you give the 4 drugs, of course, so that you're hitting this bacteria with a number of different mechanisms of action. There is a whole portfolio of drugs by different mechanisms of action under development. This is just one. I couldn't go through the whole pipeline. But to your point, in order to be ready for resistance that might develop to this, we're looking at other compounds with different mechanisms of action, and so are others around the world. There's also the rational use of medicine to make sure that there's a parallel effort working on diagnostics so that they can really determine if this is drug sensitive or is it drug resistant TB, and treating it appropriately, as we all should with any antibiotic. It's a great question. We've got to be ready for the next. And I hope that witch stays under that house.
Ash, how are we doing on time?
About 5 minutes.
5 minutes left? Okay, a few more? You're ready.
So I'm interested in this age of immediate gratification. I'm interested in timelines. How long did it take to develop this new TB drug? How long has AGS-003 taken to develop? And Lipitor and the mechanisms that lead to toxicity or not toxicity. What are the timelines that you guys are dealing with?
I'll start with the TB drug, and it's the most pessimistic of the answers because the actual the endpoint that is measured by a regulatory agency for that is 2-year relapse. That is, after you've given the drug for 6 or 9 months, you follow that patient for 2 years and see if they have a relapse of the disease. That is, did you just suppress it and then it reemerges? So that adds 2 years to the standard drug development process, which itself is already extensive. There's a partnership called Cures Within Reach that is working on repurposing drugs so that you can take drugs that have been approved for one therapeutic area and repurpose it for another, and that substantially reduces the amount of time because you have to go through a lot of the preclinical studies especially.
It's generally over a decade that these medicines require. So you start in preclinical, do animal testing, in vitro testing. Then there's the initial, you have to do dose finding, that usually takes a couple of years. Then you do dose ranging, phase 2 trials, and then a phase 3 trial is usually the longest trial that takes at least 3 years, sometimes depending on how large and how broad it is to recruit. And then based on the endpoint, if the endpoint, meaning if it's a 3-year survival trial, then it's 3 more years. And then assimilating all of that data, doing the data analysis, and getting the package ready for the FDA takes that much longer. So it is a pretty long timeline.
What about money that goes into developing a drug? I mean, I know that we saw recently a local success with the lady selling the kind of the female Viagra type of, I think like $1 billion. How much money goes into making something like that and researching? just depends, or a lot?
Well, there are various estimates. In fact, at Duke, Henry Grabowski has published a number of papers over many years, and DeMasi from Tufts, I think, in Boston. And so it's a range, but it can in fact approach $1 billion, several hundred million at the minimum.
So that wasn't a good question. All right. That's fine. All right, one more? Yes, ma'am? Okay, last one.
So, more of a question, talking about different phase of the study. We all know normally we have to do an animal study and translate that results back to human. I wonder in the personalized medicine area, which I'm not as familiar with, what kind of challenges that is we facing when transporting the results from the animal study to the human trials.
That's a really good question. I can speak on behalf of our technology and how we've approached this. So because it's autologous, right? It's a human dendritic cell, and the dendritic cell has to be driven from the patient, and then we make the therapy with the tumor-based RNA. Tried a lot of different animal models, and there are really no really good animal models for that type of therapy. There's a recent presentation that our folks from our partners actually at CellThera presented at the Society for Immunotherapy for Cancer of a mouse model where they actually implanted the tumor and then did a dendritic cell-like therapy or AGS-03-like therapy, but it still wasn't the same thing. So it's really hard to develop animal models for autologous cell therapy for human cell therapy targets.
Coming. I'm sorry. All right. Man, starting that workout early. Thank you so much. How about a round of applause? Really appreciate giving us your time this evening to talk with us. Okay, I will let you guys out of here. Just a few quick announcements. We're going to line it up for the beer line again. Okay, tomorrow, I just found out that tomorrow is National Ugly Sweater Day, Ugly Christmas Sweater Day. So, go ahead and just wear your ugly sweater, come on out. There is food truck rodeo tomorrow, I do believe. So come on out, there's a food truck rodeo over lunch hours, plenty of food trucks, tons of food. Don't forget we do this every third Thursday of the month. So January, the third Thursday, we will be doing a different topic. We rotate topics every month for those who are here for the first time. Otherwise, thank you all so much for coming this evening. Hope you have a wonderful holiday season. Get to spend time with family and friends, and we will see you next year. Thank you.